Welcome to today's webinar. My name is Jose Francisco, Project Manager at the IAS-USA. Today's webinar will be going over the role of the microbiome in modulation of inflammation and impact on disease in HIV and COVID-19. I'd like to welcome today's presenter, Dr. Nichole Klatt from the University of Minnesota. Welcome, Dr. Klatt.
Thank you so much for the introduction. Thank you, everybody, for being here today and for the invitation from Donna and IAS-USA to present on the microbiome. So the learning objectives for today, I hope that after I'm done, you will be able to explain what the microbiome is and how it can impact human health, identify the mechanisms that allow the microbiome to impact HIV transmission and pathogenesis, and describe the potential role of the microbiome in COVID-19 disease. Also, to be able to list potential interventions that can promote a healthy microbiome and decrease disease. The CME information. Support. So to navigate the webinar, there'll be poll questions with a separate window, and you can submit your questions using the Q&A button. So the first poll is, do you consider the microbiome when you treat patients, if you do treat patients with HIV, and if you do not treat patients and do research, do you consider the microbiome in your HIV research? Give everybody a second to answer that. So I'm actually impressed at 39% actually said yes, and then 60% said no. And we'll come back to this at the end. So went through this. So pretest question 1. All of the following microbiome associated factors can influence HIV transmission except for inflammation, 2, metabolism antiretroviral drugs, 3, C. difficile infection, or 4, epithelial barrier damage. So 3 of these have been associated with HIV transmission and 1 of them has not. So choose the one that has not been associated with HIV transmission. Very good. So it looks like the most people think that C. diff has not been associated with it, and then followed by metabolism antiretroviral drugs. And again, we'll come back to this after the presentation. Other pretest question: why are current therapies for inducing alterations to the microbiome not adequate? Too much colonization of probiotics, recurrence after antibiotic treatment, too many options for therapies, or bacteria can't be cultured for treatment. So which one makes it not adequate? Recurrence after antibiotic treatment is the clear winner there, 59%. And again, we'll come back to this throughout the talk and then at the very end. Great, so to get started in an actual talk. So just to make sure we're all on the same page, I think by now probably most of us have heard of the microbiome, but the actual definition is the microorganisms in an environment. This includes bacteria, virus, fungi, protists, and archaea. So we typically talk a lot about bacteria. We're going to talk about the bacteria today, but all of these microorganisms are part of the microbiome. And the genes, metabolites, and the products that they make are also part of the microbiome and extremely important and have a huge role in regulating health as well. I like to give the fun, dinner party fact that the average adult has 5 to 10 pounds of bacteria in their gut. So there's more bacteria cells in your body than human cells, and far more genes from your microbiome than there are genes from your own human cells. The word dysbiosis we use a lot, and this is when you have an imbalanced microbial community. And this can be an imbalance in either functional or abundance of the microbial communities. There's many microbiomes. This doesn't even include all of them. This certainly doesn't include the list of all of the things that they can be associated with. But just a brief overview, but virtually every mucosal surface in the body has a microbiome, as well as things like skin. Even they're talking about hair microbiomes now. So it's virtually everywhere we've looked. There's very few sites in the body that do not. We are going to focus on two in particular. I guess at the end we'll talk a bit about oral and the nasopharyngeal microbiome. And this has been associated with a lot of gingivitis and periodontal disease, even heart disease actually. And so we're going to address this a bit in COVID-19 at the end. Of course, lung microbiome has been associated with many different lung disorders or respiratory disorders, and again, might be associated with COVID-19. We're going to speak a bit about vaginal microbiome dysbiosis. This is really important in HIV transmission, as well as STIs, preterm birth. The gut microbiome is by far the biggest microbiome in the body, and this is the best studied and has been associated with the most diseases. I used to have a list of the diseases it was associated with and decided that it would be easier to make a list of diseases that it wasn't associated with, because virtually every study that has studied the microbiome in the context of any disease has found some sort of association. Extremely important in multiple diseases. Some of the biggest ones are things like colorectal cancer, diabetes, obesity, IBD, and even there's a huge gut-brain axis role in neurological disorders, and very important in HIV, and as I'll show you, and potentially in COVID-19. And there's a skin microbiome, but we won't be focusing on that today. And then just to note, there are other microbiomes. This is just a brief overview of some of them. So this is kind of my lab slide. We always kind of go back to this because there's this huge connection between systemic and mucosal inflammation and dysfunction, microbiome dysbiosis, no matter what site we're looking at, whether the gut or the vagina, and then there's immunity inflammatory responses, and all of these are really tightly connected and really important when it comes to disease. So we're first going to talk about the role of this in HIV. So as probably everybody in here knows, despite antiretroviral therapy, there's still a decreased expected survival in people who are HIV infected compared to people who are uninfected. And of course, there's increased morbidities and mortality. These increased morbidities and mortality are consistently associated with inflammation, and particularly associated with different factors that are coming from mucosal dysfunction or the gut, whether it's microbial translocation or barrier dysfunction. They're consistently associated with morbidities and mortality despite antiretroviral treatment. So we wanted to understand, and sorry this didn't come out great, at least on my screen, oops. Just an overview of potential role of the microbiome in this. And this kind of just goes through, this is a nice review that goes through the different bacteria that have been shown to be changed in HIV infection, where these little red areas are that they're shown to be up in HIV and the blues are down. And just some of them that I'll point out here are proteobacteria, which are up. Proteobacteria, it's important because they tend to be very adherent bacteria, and which means that they can grab onto the mucosal surface and tend to translocate. It also includes a multitude of potentially pathogenic organisms like Salmonella and E. coli. Things like fusobacteria are enriched in HIV infection. And again, these are going through several different papers, just kind of highlighting different papers and what they've been found. Fusobacteria is important because you have the highly associated cancers, particularly colon and anal cancer. And then you have decreases in things like rubinococci, which are very important for making metabolites that are important for bacterial health. And Then I just want to point out, and then Prevotella, which we'll talk about a lot later too, multiple studies have shown, there's a couple have shown it down, but the majority of studies have shown Prevotella increased in HIV infection. And this is important because Prevotella are probably the most inflammatory bacteria that we know of. And then just want to point out, you might be aware of the issue with, there's some studies that came out a couple of years ago showing that sexual preference was more associated with the microbiome than actual HIV infection. This is not necessarily true. There are some different changes, and there's probably more increased changes in the context of receptive anal intercourse. However, it is consistently demonstrated that even in children with HIV that are engaging in receptive anal intercourse, you see changes of the microbiome. So sexual preference can definitely have a role. As you see, it's very consistent that you see increased Prevotella in particular is associated with receptive anal intercourse and decrease of Bacteroides. And that's consistently shown in HIV infection itself. And that's, again, important because these are the biggest producers of short chain fatty acids, which is an important metabolite. Sorry, I have a puppy in the background. Hopefully she is not going to continue barking. The joys of being at home still these days. So not only, and so all those studies are showing microbiome in the gut. Let me throw her a bone. But it's also important because you have peripheral microbial diversity. So this is a recent paper from Danny Dewick's lab, and we were involved in this as well. But basically what they showed is that you can actually deep sequence in the periphery and see that there are changes in certain microbes that are actually in the peripheral blood when they deep sequence in the plasma. And then there's changes in the actual immune cells and inflammation. And this is a temporal association and can be reconstituted after ART. But this is probably associated with the microbial translocation that we see very early in HIV infection. So just to kind of summarize what I just talked about. So this is just a cartoon of what's happening during HIV pathogenesis. So in a healthy gut, you have this intact columnar epithelium, and that epithelial barrier should be intact to keep anything coming from the outside across the lamina propria. You have homeostatic cells and cytokines that maintain that healthy barrier, and then hopefully have this healthy microbiome that's highly diverse. And we'll talk about that a bit more in the context of infection. And then in an HIV infected gut, what we have seen is that you have damage to this tight epithelial barrier. These breaches allow microbes to translocate. You also have this altered microbiome, like I mentioned earlier, things like proteobacteria that can really latch on to the barrier and get through, and that's what we tend to see translocate to the periphery. You have an increase in pro-inflammatory cells and cytokines, which we'll also talk about, things like neutrophils that can then continue to break down that barrier and cause more inflammation and microbial translocation. So we really want to understand the mechanisms underlying mucosal dysfunction in HIV. So again, kind of going back to our little schematic here, one of the questions we really wanted to address here as far as this immunity component was looking at neutrophils. That's because neutrophils are extremely important for antimicrobial functions and are one of the first lines of defense. So neutrophils are actually the most abundant immune cell in the body, but probably the least studied immune cell in the body. And this is in part because they're very difficult to study. They don't survive freeze thaw. They don't make it through a Fecal with blood. You really have to do very fresh blood on whole blood to really assess these. So they're not well studied and they're not easily studied. But a big thing about these is that neutrophils are highly inflammatory. So they're the first line of defense. They get right there to try to combat a new infection. And then the way they do that is by these different roles like phagocytosis, granulation, and nets. And they release a whole bunch of antimicrobial peptides, proteases, and reactive oxygen species. The problem with that is that while it's important to combat antimicrobial functions, you need them to go away as soon as possible because they also cause tissue damage. So particularly reactive oxygen species and proteases can break down the epithelial barrier very quickly and very well. So we need to have this balance in neutrophils so that they come in, combat infection, but then they have a very short half-life, typically in hours, so that they go away and we don't have them causing damage to the tissues. So we worked with University of Washington, Northwestern UCSF to get colorectal biopsies in HIV infected ART treated patients, as well as HIV negative patients that were age sex matched. And then we got colorectal biopsies and did very quick, like I said, it's important to do this quickly, same day neutrophil counts on these. So there had really been no studies actually quantifying neutrophils in the gut from HIV infected patients before this study. So what we found here is that there is an increase in total neutrophils in the colon, and there's a significant increase in HIV infected compared to uninfected. And this is all done by flow cytometry. So we did a lot of work developing very careful panels to make sure we're getting neutrophils. And I just want to point out too that this was all adjusted. We used regression analysis to adjust for age, race, and sex and sexual preference. That is all accounted for. Now on the right here, what's important is these are cells that are longer lived. Oops, sorry, mouse is very sensitive today. So active caspase-3 low, CD16 high neutrophils basically mean these are highly inflammatory active neutrophils that are going to be robustly releasing cytokines, but they're also not going into apoptosis. So in a healthy person, when you do find neutrophils, they're going to upregulate that caspase-3 because you want it to go into apoptosis and have a short half-life. But in HIV infection, that caspase-3 is not being upregulated. In fact, so this is kind of the opposite thinking. These are caspase-3 low. So caspase-3 is an apoptotic marker. So we wanted to understand, well, what's going on? Why are these neutrophils not dying? Why are they not going into this short half-life so they don't cause tissue damage? And I know it's kind of a confusing plot, but the important thing here is that what we see is there are changes to the microbiome in these patients, and this is a very small cohort. So even it's 23 and 25 patients in each group, and we still see differences of the microbiome. And again, this is all after regression analysis to account for sexual preference and different confounders. But importantly here on the right is that when we look at, so Lactobacillus is a very good bacteria that typically used as a probiotic. We'll talk more about Lactobacillus shortly. And then Prevotella is this highly inflammatory bacteria that we know is increased in HIV. And when we look at this Lactobacillus to Prevotella ratio versus this neutrophil survival, we see a significant negative correlation indicating that maybe there's a role for the microbiome in this neutrophil survival that we're seeing that might drive these neutrophils to be accumulating in the gut in HIV infected individuals. So I know, again, this is a very complicated slide, but just quickly wanted to demonstrate that then we can go in vitro. And what we did here is we took neutrophils, we took whole blood from patients, and then exposed it to different bacteria to see what induced the caspase-3 and neutrophils, so the apoptosis. So on this one, we're looking at, we want to see high caspase-3, we want to see these cells going into apoptosis. So with no stimulation, you see a lot. Lactobacillus is good, but it actually induces more apoptosis, which then will allow those neutrophils to go and die. But then when we look at these different bugs that are increased in HIV infection down here, they actually decrease apoptosis. So this can actually demonstrate, so we saw this association in vivo and then go to the lab in vitro, and we can validate that we can actually induce this phenotype of long-lived neutrophils and decrease apoptosis by exposing them to bugs that have been associated with increased or that are increased in HIV infection. So just to conclude this part, neutrophils are increased in the GI tract of treated HIV infection and associated with decreased neutrophil apoptosis, which is what we think the mechanism is for the accumulation of neutrophils in the gut in the context of HIV. HIV-altered mucosal bacteria differentially affect neutrophil apoptosis, and where you have decreased lactobacillus or good bugs and increased prevotella or bad bugs, that might prolong neutrophil lifespan in the GI and treated HIV infection. But we really are working hard to further understand how neutrophils contribute to HIV pathogenesis and barrier damage to potentially elucidate targets for therapeutic interventions. So I like this slide kind of highlighting that there's these overlapping mechanisms of pathogenesis and transmission. So obviously we just talked about pathogenesis. I can show you that I hopefully have convinced you that an altered microbiome, one role can cause of that, it can be inducing inflammation through inflammatory cells like neutrophils that might lead to barrier damage because they're releasing proteases and then can affect that barrier and cause microbial translocation. But there's similar roles in transmission that we're going to talk about now. So I always highlight the importance of studying women in HIV. So often in the US, we think of the burden of infection is in men, particularly in gay men. But in sub-Saharan Africa, we have 2 times the amount of women ages 15 to 24 than men in the same age getting infected. And there's actually now more infections in women worldwide than there are in men. It's also important to study women because obviously the pregnancy and mother-to-child transmission issues, and especially in areas that are not getting good ARV availabilities. And then of course, there's the issue that in a lot of the areas with the highest HIV rates in women, you also have sexual violence and lack of condom or sexual protection negotiation rights. So these women can't actually ask to use condoms. They don't have that right in a lot of these countries. So we really want to understand in my lab what the biological mechanisms leading to HIV acquisition in women are. So again, one of my little cartoons here on the left, you have a healthy female reproductive tract where you have you want in a healthy female reproductive tract, you want an intact squamous epithelium here. You have, again, homeostatic cells and cytokines that regulate that barrier and help protect from pathogen infiltration. Now, we'll talk much more in depth about the female reproductive tract bacteria, but Lactobacillus is what we want dominant. So the female reproductive tract is opposite of the gut. In the gut, you want more diversity and higher diversity to be healthy. In the female reproductive tract, you want lower diversity, you want just Lactobacillus. But there's several putative mechanisms that have been associated with increased HIV transmission. So this includes an altered microbiome, which we'll talk much more in depth about. This can increase the pH and cause barrier trauma. You have damage to the epithelial barrier, of course, can let the virus go through. And similar to the gut, you have increased pro-inflammatory cells and cytokines that can be targets for infection. And as well as things like neutrophils then can release proteases and break down that barrier and increase infection. So this is kind of the 30,000 foot view of dysbiosis in the context of the vagina. So like I said, in the optimal case, you want dominance by Lactobacillus, particularly Lactobacillus crispatus. Oops, sorry, this is on a timer, I guess. And this will keep the pH low, which is very good in the vagina, and that can protect from pathogens. So clinically speaking, this is diagnosed by Nugent score. So you would see a wet mount like this that you see there's all these lactobacilli, very few cells, and then this would be associated with a low pH and would be Nugent negative. In the context of bacterial vaginosis or dysbiosis, you have an increase in polymicrobial anaerobic bacteria such as Gardnerella, Mobiluncus, Prevotella. Sorry, I don't know what this timer is doing here. And Microbiome. This is associated with increased pH, increased inflammation, increased barrier damage. And then when you look at the clinically scored Nugent score here, you have a loss of the lactobacilli, but lots of polymicrobial flora and these big clue cells. And then you have a much lower pH or higher pH, I'm sorry, and that's going to be associated then with a Nugent positive or what would be diagnosed clinically as bacterial vaginosis. Sorry, I have a very crazy dog in the background. I hope that you guys can't hear her and she's not distracting. Puppies, COVID puppy. So this is how we tend to look at the actual bacteria. So the Nugent score is kind of done clinically, but it's not the best measurement because it's very subjective. You have different nurses or clinical staff that is reading it and scoring it. But of course, then we have the sequencing version of looking at bacterial vaginosis or dysbiosis. Now, these are data from Doug Kwan's lab in the FRESH cohort, which is a cohort of young women in South Africa. And just to orient you to this, each line represents one woman, and this is cervical vaginal lavages. And each color represents different bacteria. And then he broke these down in different community type structures where you have CT1 is going to be your optimal community, which is Lactobacillus crispatus. here, and that's going to be your kind of best ideal bacteria. Lactobacillus ginsengii is another one that falls into this, but you rarely see this in Africa. CT2 is Lactobacillus inners, which even though it's a lactobacillus, this is kind of your worst lactobacillus, and it's often associated with a transition into BV. And then you have CT3 and 4, where you can see that you get a lot more diversity, and this is often associated with a lot of Gardnerella vaginalis, which is in blue. I'm just going to throw another ball to my dog. Sorry, I was hoping she would be under control today. So the reason this is important is when you look at by these community type structures at HIV infection risk, you have 0 infections in that CT1 group, or the Lactobacillus crispatus. However, as you increase even to CT2, Lactobacillus inners, and then diversity in these CT3 to CT4, you see increased HIV infections. Also, in a meta-analysis study with Bacterial Vaginosis, and this was all clinically diagnosed Bacterial Vaginosis by Nugent scores I showed you before, even though it's not the best indicator of it, there still is, you see this high increase in HIV transmission in women who have bacterial vaginosis. So you have 3 times actually increase in transmission of women who have bacterial vaginosis. And I always like to point out here too, it's not just important for women with bacterial vaginosis, it's also important because men who are sleeping with a woman that has bacterial vaginosis also has a 1.8 increase or 1.8-fold increase in HIV transmission. And it also increases mother to child transmission. So bacterial vaginosis can affect all populations in HIV transmission. So it's really important to understand this. Furthermore, just like we showed you with the in the gut with this increase in these inflammatory neutrophils, we see that there's these phagocytic and inflammatory neutrophils that come in the context of bacterial vaginosis. Whereas these good neutrophils that we want to kill bugs are decreased in bacterial vaginosis. And again, we see increased neutrophil survival and increased overall neutrophil count in the context of bacterial vaginosis. So very similar to what we see in pathogenesis. Again, this is in uninfected women, but bacterial vaginosis might be bringing in these inflammatory neutrophils, causing inflammation and barrier damage, and allowing the gut to get through. So kind of the model that we like to propose for how this happens is that you have this nice lactobacillus in a ideal environment with homeostatic cells and cytokines and an intact barrier. But when that lactobacillus is replaced by diverse microbiota, so this is in the context of bacterial vaginosis, then that brings in these neutrophils. The neutrophils release all these proteases and can break down that barrier, causing barrier damage. They also, part of it, when I talked about these MDCs, these suppressive cells, they induce PD-1 expression on CD4 T cells. This can, and then that barrier damage can allow the virus to get through. And these are preferential targets for HIV infection, highly activated cells that they're activating. So this is kind of our proposed model for what we think is going on as far as an inflammatory role. But we wanted to then move on to try to understand the role of bacterial vaginosis in PrEP. And so pre-exposure prophylaxis. So in men, PrEP is highly effective, where you see 89% efficacy in PrEP trials, where in women you have extremely more variability from negative 49% to 75%, and even that 75%, these top 3 here, 66 to 75% up here, those are in women who were in a discordant relationship, so they knew their partner was HIV infected, and they were aware of it. And so even in the best case scenario with them when they're aware of it, we still see lower efficacy of PrEP than what we see in men. And this is a couple years old now. There have been some recent trials. But even in some of the best trials that are moving forward, like dapivirine, you still only see a 30 to 40% efficacy. So we just are not seeing the same efficacy in women as we see in men. So many people have attributed this to adherence, which we never totally could swallow that just women were just worse at adhering than men. So we really wanted to understand if there's biological factors that affected PrEP. So we worked with the Caprisa 004 tenofovir gel PrEP trial. So this was a tenofovir gel that was inserted vaginally. So it serves as a microbicide pre or post coitus. Their overall study back in 2010 that did this showed that when you just compare all women, placebo to the tenofovir gel, you had 39% efficacy in this trial. So we took samples, cervical vaginal vages, and I did this in collaboration with Adam Bergener, and we took samples and looked at the microbiome in these women. For power purposes, we just split these into two groups. So women who are Lactobacillus dominant in orange, or women who had this polymicrobial flora, and again, that's associated with a lot of Gardnerella vaginalis. So we really just call them Lactobacillus dominant or not Lactobacillus dominant. And again, to remind you, Lactobacillus dominant is the ideal, what we want, right? So then once we split women into these two groups and then looked at efficacy again, we were absolutely shocked to see that women who have Lactobacillus dominant vaginal microbiomes, the efficacy goes up to 61% for the tenofovir gel. But women who do not have Lactobacillus vaginalis dominance, they go down, the efficacy goes down to 18%. So you lose efficacy in a huge way if you don't have this ideal microbiome. And this was the first time that's been shown that the microbiome is associated with differences in efficacy in a PrEP trial. So we went on to assess, we wanted to, and in that original study we published in Science a few years ago, we did then go on to show that tenofovir could be metabolized by Gardnerella and different dysbiotic bugs. But we have since gone on to study this more intensely using, and we did that in a lot of co-culture cells, but now we are assessing biodegradation of different drugs by microbial communities using primary samples. So we take CVLs from women that either do or don't have BV, and then we look at the potential for these bugs to biodegrade or metabolize different drugs. So we're looking at tenofovir, since that tenofovir gel was that first trial and has been used in a lot of these the microbicide trials, but we also went on to look at TAF. So of course, as most people here know, TAF is next generation tenofovir and is used now in a lot of PrEP regimens, particularly in men, not so much in women. And then dapivirine, which has been used in the dapivirine rings in women. So they have so similar to like the Nuva ring for birth control, they put the dapivirine in, and then the ring sits in the vagina. And of course, this becomes really important for if there's constant exposure to these microbial communities. And then, like I said, we took samples from women with or without BV, the cervical vaginal lavage or CVL samples, and expose them to these drugs in the context and put in target cells to determine the potential for these bugs to metabolize these drugs. So here, the way we're looking at this is, again, remember each line is a woman and each color is a different bacteria, with the orange being our Lactobacillus, our ideal, and then you can tell these different polymicrobial are going to be our BV or dysbiotic states. And the way this is ordered is from left to right is the least degradation of the drug to the highest amount of degradation of the drug. And what you can see for tenofovir here is it's very clear that there's less degradation when there's Lactobacillus dominance and more degradation that keeps increasing with the diversity of the flora. And then this is just on the right-hand side here where we actually quantified the degradation so this is tenofovir diphosphate. So this is the pharmacologically active form of tenofovir that would be in the cell and needed to be actually active and protect against HIV infection. And what we see is that when you have less than 50% lactobacillus, so these are the BV states in the purple, you have much less active drug, and that the rate of degradation is associated with how much lactobacillus, or is negatively associated with how much lactobacillus is there. When we look at dapivirine, again, this is important because these dapivirine rings are sitting in the vagina, constantly exposed to these bacteria. And again, we see the same thing from left to right, where you see very little degradation if there's lactobacillus, and the degradation increases with the microbial diversity. There's, you can point out there's clearly some association here with the Shuttleworthia, which I should mention, it might not be, I think that might be a misclassification. There's a huge debate in the microbiome field about what this bug actually is, but we know it's a bug associated with BV, and it's unclear exactly if that's Shuttleworthia or not. But that's just if there's any, microbiology people in the audience would notice that, but nothing to focus on. But anyways, the same thing here. When you look at the women who do not have lactobacillus on the right in purple, you have less of the active dapivirine, that intracellular. If you have BV, whereas you have lactobacillus dominance, you have a much higher rate. And again, that rate of degradation is inversely associated with lactobacillus. But then when we look at TAF, so the next generation, tenofovir, you see that there's no kind of, correlation like we saw earlier. There's no trend here. And that's because the TAF is not metabolized. We don't see TAF metabolized in any of our culture systems. So this is, and then, accordingly, there's no association with the rate of degradation. So this is very good news that there are drugs out there that aren't metabolized by vaginal bacteria. And so this should be really considered for PrEP. So just going back to our little cartoon, I think that, the algae microbiome can increase inflammation and barrier damage. We've also done a lot of work. I didn't have time today to talk about the barrier damage that algae microbiome can directly induce. But also this ARV drug metabolism is another thing that we really need to consider, because this could be having a potential role in decreasing PrEP efficacy. So the second conclusion here is dysbiosis of vaginal bacteria is a key factor in vaginal inflammation, barrier integrity, and HIV acquisition. And dysbiotic bacteria can metabolize the PrEP drugs tenofovir and dapivirine and potentially contribute to decreased PrEP efficacy. But TAF is not degraded. So again, this could be a consideration for efficacious PrEP. And we should really consider drug metabolism as one of the factors that when we're actually testing new PrEP drugs, especially in women. But most importantly, we need to better understand the vaginal microbiome and how to increase lactobacillus communities and prevent BV and dysbiosis. And I should have mentioned this earlier, and we'll kind of get back to this at the end when we talk about therapeutics. But one of the big problems with bacterial vaginosis is, even though you have 60% of women worldwide that have bacterial vaginosis, there are very few treatments for it. You have 2 different antibiotics and only 2 antibiotics that are clinically approved to be tested to be used. And they have a 90% recurrence rate after a year, 50% recurrence rate after a month. So they do not work, and we need better treatments. So kind of going back to my little schematic here, we're doing a lot in my lab with all of these different things. And really it's, when we're working on multiple different platforms and diseases, but where this whole, kind of interchange of the inflammation, mucosal dysfunction, microbial dysbiosis, immune inflammatory responses all are associated. And now, we're adding kind of this factor of drug metabolism into it. This is important. No, I'm sorry, this figure didn't come up well, but we are not the only ones who've looked at this drug metabolism. It's actually been studied for many, many years. The drug field or the pharmaceutical fields have been looking at this for years, and there's a lot of old data out there that have shown that bacteria can metabolize different drugs. But there's a lot of novel work coming out. So this is from Andy Goodman's lab, who did a beautiful screen where they actually looked at 300 different drugs and found that 90% of the drugs that they looked at in these screenings could be metabolized by different gut bacteria. So we're moving on in my lab to understand, so I showed you all that in the context of the vaginal microbiome and PrEP, but both for oral PrEP and for HIV infection, and looking at reservoirs and how the antiretroviral drugs might be degraded and then contribute to increased reservoirs and lack of being able to cure HIV. We're trying to understand gut bacteria and the role of metabolizing antiretroviral drugs. We know just tenofovir and stool, and brown here, we can see that there's degradation when we do daily, we daily dose tenofovir. So it would be like the same thing as if you're taking PrEP every day or your drugs every day. And again, we are moving on to newer generation drugs we have underway in the lab that are not tenofovir, but that are more widely used. But we do know that tenofovir can be metabolized by a multitude of different bugs that are in the gut, shown here and shown over here. So this is something that could really affect oral PrEP and could affect, just in the context of HIV infection and pathogenesis. And so we're working hard in the lab to try to understand this. And this is kind of the overview of what we think might be happening, is that, we have a multitude of things that microbiome dysbiosis is associated with that could impact the HIV reservoir, but, immune responses, inflammation, of course, but also, microbiome-driven ARV metabolism can affect that proteome and the ARV drug levels. So that is ongoing work in our lab, and maybe I'll come back in a year and tell you more about that. So back to kind of schematic, then we wanted to go into COVID-19. So obviously all of us working on HIV had to quickly shift gears in the last year and think about COVID. And so we wanted to understand if microbiome was impacting COVID-19 disease, and could it be one of the mechanisms underlying disease severity? And we're working hard on understanding if pre-existing microbiome contributes to disease outcome in COVID-19. There is some data. So this is from Michelle Newsom-Swog's Nature paper a few months ago, where they actually show that you have long-term SARS-CoV-2 replication in the gut. And there's also been a lot of gut pathology seen in long-term COVID and during COVID, active COVID cases. So reason to believe that there might be something going on in the GI tract. There's also been a couple of studies that have shown differences in SARS-CoV-2. This is showing longitudinal patients and changes to their gut microbiome with SARS-CoV-2. And so this is a nice summary of these longitudinal patients, but it's a very small cohort. And another study, the oropharyngeal microbiome was shown to be different in COVID-19, and diversity, which again, the diversity is very important measurement of kind of overall health of the microbiome, and showing that in critical patients with COVID-19, you have decreased diversity in the oropharyngeal microbiome. So reason to believe that there could be a role for the microbiome in SARS-CoV-2. So I'm going to show you data from two studies. One is, so very early in the SARS-CoV-2 epidemic, the COVID-19 epidemic, we got samples. So I have a very talented assistant professor in my group, Luca Schifanella, who trained in Milan. And so he got his MD there in infectious disease, and so reached out to his colleagues there. If you remember, back in last March and April, Italy really got hit hard with COVID-19. And so we got rectal swabs from individuals during March and April of 2020. These are all hospitalized patients, all confirmed COVID. There was one time point, so they took rectal swabs at one time point through them. We had 96 patients, 96 rectal swabs, and we did 16S rRNA sequencing. We had 33 females and 63 men in this, and the median age was 59, so actually pretty young. And our primary objective here was to investigate differences in the taxonomic profile of the microbiome across clinical outcomes. So the biggest clinical outcome, of course, is people who were deceased and got better, or deceased and obviously died, or the people who got better and were discharged. And this is just showing you the top 10 phyla. And what you can see, and this is the average on this, is there wasn't huge differences. So, and just when we look at deceased versus discharge, we did not see major differences in the phyla, nor did we see this in the genre, looking at a lower level. And then when we look at the diversity measurements, and again, this is all rectal swabs, so gut, when we just look at deceased versus discharge, and we've looked at a lot of other factors as well. So we've split this up by age, we've split it up by people who are intubated or not, a lot of different clinical outcomes, and we really haven't seen anything highly significant. So once people are hospitalized and at that one cross-sectional time point in this cohort, we really didn't see much for changes. So then we also had a biobank here at the University of Minnesota, where we took nasal, oral, and rectal swabs collected, and people had an option of if they wanted to do all three or one of the three. So the numbers aren't all the same of how many we have. So we had 20 rectal swabs, 65 oral swabs, and 62 nasal swabs. But these were all hospital patients confirmed with COVID. Again, one time point taken. We had 41 females, 53 males, median age of 63. And this, again, all was in Minnesota. So here we have nasal swabs on the top left, oral swabs over here on the top right, rectal swabs in the middle here. And again, we broke this up into deceased versus living. And again, we saw no major changes, no significant differences in any of these microbiomes in patients that were hospitalized here in Minnesota with COVID. There were some minor changes. Oops, sorry. Where this goes, but nothing was significant when we looked at overall phyla or genus. Now again, looking at the diversity, when we look at the rectal swabs, we didn't see any differences in the diversity. And so the richness is just the number of bacteria where the evenness is the bacteria relative to one another, and then the Shannon diversity is just overall diversity. So you do see that there's trends in deceased to be a little lower in the rectal, not so much in the oral, but none of these were significant. However, what we did see was that the people who were living versus deceased in the nasal swabs, there was a significant increase in the richness. So again, this is the overall number of bacteria. So potentially something there when we actually look at nasal swabs at a cross-sectional study here. This is actually looking at relative abundance of specific bugs that then came up significant. So on top we have 2 from oral swabs. Oops. Sorry, so we have fusobacteria that was significantly lower in the deceased, where we have megasphaera that was significantly increased in deceased in oral swabs. And then the Sphingoldia that was significantly decreased in the deceased from the nasal swabs. So there's a couple bugs that are coming out, we're following up, and a lot of these data are quite new. So we're still doing a lot of work here, trying to kind of sort out what this might mean and if it does mean anything, and following up to see if we can consistently see this in bigger cohorts. So I know it's not very exciting data, but we have a lot of work still going into this. This is really, some of this data just came off the sequencer like a week ago. So while we didn't see any huge changes in the microbiome based on the outcome from these small COVID-19 cohorts, by 16S sequencing, there could be potential species-specific differences that we need to follow up more. And we really need larger cohorts with more comprehensive data and longitudinal sampling. And so that's something that's going to be critically needed, especially if this pandemic stays around, which it seems like it's going to for a bit. And again, we're doing this whole genome sequencing is underway, and all of these samples for further analysis. And we've been working closely with groups like Rob Knight's group, who has some very interesting data on some of the actual functionality of the microbes that are associated with COVID-19 disease. So, and we'll be able to get a much better view of it as we're doing these deep sequencing of whole genomes. So finally, I want to talk about how we alter the microbiome to improve health. So I really love this slide because it kind of shows, you want this flourishing gut ecosystem or whatever microbiome ecosystem it is, but then you give it antibiotics or you get sick with something or, whatever it is that might alter that and devastate it. And then, if you don't actually treat it and help it, it'll be, species run wild and you might get things that you don't want, like dandelions. So, there's different kind of the 3 main therapies that have been used and are routinely used. You have probiotics. So probiotics are basically your good seeds, right? So you want to give it like that best grass seed. And probiotics are typically Lactobacillus, Bifidobacter. There's some yeasts that are used. Lactobacillus is by far the most common. And that's just giving good bugs. There's a lot of issues with probiotics. There's, very differential data on whether they do work or not. They rarely colonize. You have to take it every day. I always say that probiotics will never hurt. And I think a lot of the probiotics don't get through into the gut and are, and they aren't alive to colonize, but there's still, I think, often, good metabolites in there that can help. But it's not a magic bullet by far. Prebiotics can help, and in combination with probiotics, it can be even better. But prebiotics are basically food for the probiotics. And these are things like fructooligosaccharides or soluble fibers that can just basically promote good microbe growth. So, different bugs like to eat different things. For example, there's a lot of bad bugs that like to eat, fats and carbohydrates, so they've been associated with obesity, whereas in these good bugs that we want tend to eat more high fiber and fructooligosaccharides. So giving these prebiotics might promote better growth of your, good probiotic bugs. And then, of course, we have bacterial therapy. So this is transplant. So we've probably all heard of fecal transplant. So like in the context of C. diff infection, fecal transplant has been extremely effective in curing C. diff infection. And that's transplanting the whole ecosystem. However, that, while it's been working in things like C. diff, it's been shown in HIV to have, differential results, nothing very compelling, however. And then in things like IBD, it's been very disappointing that it hasn't worked. But, ultimately, we want to get back to this good ecosystem. So there we go. So There's other mechanisms we can think about to target that are kind of coming up the pipeline, but we really need to, I think it's really critical for the microbiome field to get better ways to adjust the microbiome, because it's really just, we don't have anything great now. So on top, just as a side note, with the vaginal dysbiosis, as I mentioned, you have metronidazole and clindamycin are the only options for BV. Metronidazole is the first line, and it's also a horrible antibiotic that has a lot of side effects. And it just doesn't work. You have frequent recurrence. We have an ongoing study where we're seeing up to 70% recurrence within a month. So it's not good treatments. We need better treatments. But in general, even for the gut, for anything, we need better microbiome therapies. Some of the things coming down the pipeline, targeted probiotic therapy. So really having kind of personalized medicine, looking at ways we can target specific probiotics that will thrive in a better environment. There's also engineered probiotics or live biotherapeutics. An example of this is Lactin V was just the New England Journal paper just this last year showing the effects of Lactin V. Sorry, bad puppy. In dual treatment with metronidazole and having a better effect in enhancing BV. However, again, it doesn't work in everybody. We need better biotherapeutics. Bacteriophages is another one. So bacteriophages are actually viruses that can go in and kill the bacteria. So for example, Stephanie Stradi at UCSD is running a bacteriophage therapy center that they've been really doing amazing work and using bacteriophages to target specific bacteria. But again, you have to be able to find a specific bacteriophage to get in and target those specific bacteria. So this is still in the early stages, but very promising. You have the microbiome material transplant. So whether it's a fecal transplant, or there's been some recent work looking at vaginal fluid transplants, or an Elanov in Israel has published a couple of nice papers that have shown some efficacy in treating BV by using, women who have lactobacillus and actually taking CVLs from those women and transplanting them into women who have BV. You can also do things like bacterial ligands or metabolic components. So rather than using the actual bacteria, like, can you just stimulate the immune system or, increase homeostasis by using those ligands? Targeted antibiotics, which is a new antibiotics is a newer field as well. And then metabolic components. So, a lot of women use boric acid to try to treat BV, but there's a lot of different things that are looking at in the gut as well, like bile salts and sialic acid. So there are options, there's things coming up, but there really needs to be better and more research in this. So just want to thank my lab quickly and my collaborators, Adam Bergener, who's now at Case Western, and Caprisa. And with that, we will go back to our questions. So again, the post-test questions, same thing I asked you earlier. We'll see what we got for the answers now. So all the following microbiome associated factors can influence HIV transmission except for, so hopefully we went through a lot of these and you guys will have known what the answer is now. You did pretty good on this one the last time. All right, so C. diff infection is the correct answer here. So C. diff infection has been associated sometimes, that it's been shown in a couple of studies that it's increased in HIV pathogenesis, but it has never been shown to be increased in HIV transmission. And a few of you still think metabolism antiretroviral drugs, and hopefully I showed you that in our studies that the PrEP drugs we showed were metabolized by the dysbiotic vaginal microbiome, and so that might affect HIV transmission in that way. So post-test question number 2, why are current therapies for inducing alterations to the microbiome not adequate? So again, so which one is not, or which one is the reason that they're not adequate? So is it because there's too much colonization of probiotics? recurrence after antibiotic treatment, too many options for therapies, or bacteria can't be cultured for treatment. So which one is not, or which one is one of the causes for the therapies not to be adequate? Very good. So recurrence after antibiotic treatment, that is exactly right, as the majority of you learned. So for bacterial vaginosis, you have huge recurrence after treatment, but even in the gut, which I didn't show data on this, but we do see that after antibiotic treatment, after about a month, you're back to the pre-antibiotic state. So recurrence after antibiotic treatment is a huge problem for why antibiotics can't be used to alter our microbiome, or have not in current, have not been used well. Oops. So going back to the original poll, after this talk, we now consider potentially the microbiome when you treat patients with HIV. Well, great, 93% of you I have convinced that it might be something important. Again, unfortunately, the bad news is it's hard to change, but there are things like probiotics that, I always kind of say they won't hurt. And there are some ACTG trials that the data are coming out more and more they did a big probiotic trial. So hopefully in the coming years, we will have much better therapeutic options to go into. So with that, I am happy to take any questions in the audience, and I apologize again for a cranky little puppy today. Sorry, I'm trying to pull up. There we go in the Q&A. From Axel Vanderpeer, sorry if I butchered your name there. What kind of probiotics could be useful for HIV positive patients? So there's a lot of probiotics out there. And I'm no expert, and I'm of course not an MD, I'm a PhD. But I've done a lot of work on Dysbiosis and VSL3, which is, sorry, things are moving around in the background for me. Those have been, and that's what was used in ACTG trial. So we did show some good effects of that in SIV infected monkeys back when I was in Jason Brenchley's lab. And that is available online. They're not available in stores. As far as over the counter in stores, Culturelle seems to be a good product. They have Lactobacillus rhamnosus or Lactobacillus GG. Is anything that includes that has been shown. There's been a lot of clinical trials on that. And then there's a couple of yeast products with Saccharomyces that have shown to have some good effect, but there's not a lot of clinical data out there. Catherine Kripke asked what causes vaginal dysbiosis. Another great question. So this is unclear. There's been a lot of association. So there's certainly a factor of hygiene and intravaginal practices. Douching is highly associated with increased vaginal dysbiosis. If you see a lot of women, I highly recommend that you tell them not to douche. Douching is consistently associated and is very cultural. So we see it, especially in African-American women, they like to douche, and in certain populations, and this has been associated consistently with increased bacterial vaginosis. We're also working hard in my lab to try to understand this better and the root cause. But there are genetic factors that seem to be associated, and we're working on some single cell RNA-seq assays and really kind of getting a deep dive into why some women, have pre-existing vaginal dysbiosis and don't. Rob Gordon said, didn't H1508 show high prep efficacy with IM cabotegravir in cisgender women? Yes. So I had mentioned that there's been a lot of newer studies that show better effects of the of newer drugs for PrEP in women. And of course, if you're giving something IM, you're going to be bypassing that the vaginal dysbiosis issue. So I'm very excited about some of these new IM therapies. But again, there's issues with those. They're not going to always be available worldwide, and not everybody's going to want to get a long acting. And it's also if there's any allergies. So, we still need other options, but this is definitely the most exciting kind of next generation of this, that hopefully we can bypass the entire antiretroviral degradation by bacteria by doing things IM. So Katrina Nugan, such an increased role in the microbiome. What is the role of vaginal douching? I think I kind of just addressed that. How can patients bypass the odor without douching? So yeah, it's hard. So and douching actually can increase the odor. It seems counterintuitive, but while it might have a temporary effect, then it also promotes more of the bad bugs. And so that can increase the odor long term. So boric acid supplements have been shown to help. There's a product on the market called Refresh that I know has been, it's a gel that, and I should know, I don't remember offhand what is in it, but it's a pH reducing gel again, and that seemed to be more effective. I would recommend, instead of douching, trying one of those suppositories, which might be beneficial. There's also 2 over-the-counter products on the market right now. There's a cultural for women that has Lactobacillus ginseni and Lactobacillus crispatus in it. And then there is it's a major, I think it's called AVO, is the other company that they do a lot for UTIs, but they also have the actual vaginal lactobacillus. But you have to watch out. And so there's a lot of products in the market that say it's aimed for vaginal health, for probiotics, and almost all of them are gut lactobacilli that you will never find in the vagina. So if it says like Lactobacillus plantarum or Lactobacillus rhamnosus, these are gut bacteria. So it's really important, as we're treating women with these probiotics, that they actually have Lactobacillus crispatus or Lactobacillus ginseni, or both in them. And those are the only 2 products I'm aware of. Really hoping that Lactin V, I think, is the most promising. It's Lactobacillus crispatus suppository, and I'm really hoping that will be on the market and available and FDA approved soon as a live biotherapeutic, because I think that's more promising than anything currently on the market, unfortunately. Does oral lactobacillus therapy correlate well with vaginal lactobacillus concentration? So there's very limited work, and that's from Jay Chang, very limited work on this. There was a study by Lowell Leganair several years ago showing in monkeys, they have this beautiful lactobacillus that kind of lights up blue that they actually could find if you gave it to monkeys orally, that they could find it in the vagina. Of course, monkeys are a different situation though, because they also have not the best hygiene and tend to put stool in the vagina. There's not a lot of data on women on this. And it's kind of conflicting. So I think the jury's still out on that one. There's definitely associations, and it definitely can occur, but it's not consistently been found. Annie Elbury, can you talk about probiotic vaginal suppositories using EU and Canada? How effective are they? Yeah, so this is another thing, I was actually in Germany and I gave a talk on the vaginal microbiome, and someone had said, well, we have really great suppositories here. And she had, gave me a list of them, came afterwards and gave me a list of what they were. And I went to the pharmacy, and like I said, they're all gut bacteria. So none of them are gut, they weren't Lactobacillus crispatus, Lactobacillus plantarum. They were like things like Lactobacillus plantarum and rhamnosus that don't make the correct isoform of lactic acid that you need in the vagina. So I don't know, and I don't know if there's been very good clinical trials showing it. I'm not familiar with what they're using in Canada, but I do know, as far as in Germany, and I also looked at these, I was in Zurich and went to a pharmacy to see the same thing. And again, these are not actual vaginal bugs that are the ones that are highly used there. They're actually gut lactobacillus strains. So I'm not very confident that they would be highly effective. Might be better than nothing, I don't know, but I don't know of any actual clinical research that's been done to study that. And Wagon asked, has there been any research regarding histamine intolerance that can develop from overabundance of probiotics? And unfortunately, I do not know the answer to that question. I can certainly look into it and get back to you if you want to send me an email. but I am not sure. I know there's been, probiotics are very well tolerated. Typically, there's been a couple, case reports of somebody who's gotten bacteremia afterwards, but these tend to be people who are extremely immunosuppressed and already have very dysbiotic microbiomes. And even that, it's association at best, and they're not showing outgrowth of the actual probiotic strain as much as they're showing outgrowth of other strains that might not be associated with the actual probiotic. Oak and Eric, there are patients with antiviral resistant HIV. How can a person consider the microbiome? Let me say that see if the desired combination of therapies disrupts the microbiome. Yeah, so two kind of questions and good points here. So there is this kind of opposite effect. So I talked a lot about how the microbiome might affect antiviral treatment, but there's also data showing that the antivirals themselves can affect the microbiome and change the microbiome. So there is that to be concerned about. But this is one of the things that we're really trying to get to the bottom to in our lab, is understanding, are there certain bugs that are associated with antiviral resistance, or that have, increased metabolism of certain drugs that we need to monitor patients for personalized medicine. So, the jury's still out. We're working on this right now. I'm not aware of anybody else who's looked at this and has actual data on this, but it's a really important question that we're trying to address. Adjust Joseph, what is the preferred diagnostic test for BV? So that's a good but loaded question. So Nugent score and Amsel's criteria kind of showed you these wet mounts. That is classically used clinically. We have shown that up to 50% of women who would come up BV negative by that actually have BV by sequencing, when we actually go in and sequence accurately. But obviously we can't just do, deep sequencing on every single woman who's come in. The pH is the most highly correlative of looking at sequence diversity versus a clinical test. pH is typically highly associated. If you have a pH under 4, it's almost always going to be Lactobacillus. So that's good. And that is kind of in combination with Nugent's and Amsel's. But it's better to have a non-subjective test. There's a couple of things. I just read an article the other day in The Economist of a student from Stanford that's developing a more rapid test, a rapid diagnosis test that women can even do at home. So hopefully there's going to be more stuff. But women's health is vastly understudied, and there really a lot to do here, and a lot of work that needs to be done to be able to better diagnose clinically and in the field. And so and this has to be considered too in clinical trials. And I always say, especially in these clinical trials in South Africa with these young women where you have 60% rates of BV, if we could have a rapid test for them to say, at the time they come in for their prep, hey, you have a particularly bad microbiome, you need to be particularly careful because this prep isn't going to work as well for you. And have more counseling and potentially say, give them boric acid or do something that, hopefully these better live biotherapeutics will come out and better be able to treat and prevent the BV and let women know they might be at higher risk because they have it. I think would be really important, and we're not quite there, but hopefully working towards that. Privilege Manjama, we're expecting the rollout of the depivirine ring soon in our country. If it is affected by the vaginal microbiota that much, won't using it for a month as recommended be a big risk for HIV transmission. Do you have any recommendations regarding this? So this has been a bit of a issue with this. So the people who've done the depivirine ring trials claim that they have not seen any differences. They did do measurements by a Nugent score, I believe, and they do claim that they haven't seen differences in women who have BV versus who do not with the vaginal ring. Given our data, I am a little concerned, but I think we need to. I've been trying to get samples to be able to actually study the direct samples from women using the depivirine ring and the microbiome. I have not had those available to my lab as of yet, but we are working hard to be able to understand that question. And hopefully, because you have it there every day, hopefully that daily release of the depivirine will overcome the microbiome. But again, I think that we just don't have the actual scientific data on that yet to say in vivo, because again, all the data I just showed you was from, using primary human samples, but, and then giving the drug ex vivo. So we really need that in vivo data. And we are working on trying to get those samples to answer that question. Maggie White, is there any data looking at the effect of anal douching, i.e. enemas and flushing on an MSM population on the microbiome? Excellent question. Actually, one of the companies I consult for, Future Methods, is working very hard on making a better anal douche. So there is associations with douching and dysbiotic microbiome, including increased fusobacteria, which is highly associated with anal cancers. And this is another problem, right? So anal douches mostly are just fleet enema right now. There's not options for people who want to use anal douching. And so I'm really excited working with this company, Future Methods, that's making these anal douches that are going to enhance the microbiome, and they actually are putting probiotics in it, and hopefully enhance that epithelial barrier. I think this is really important to have products on the market that are going to be better, because it's fleet animals. Not only can they alter the microbiome, but they can also actually disrupt that epithelial barrier and increase the chance of HIV transmission. Dwight Ferris, any consideration of altering the microbiome of the sexual partners to help reduce the recurrence of vaginal dysbiosis? That is a great question, and there are associations with the penile microbiome and vaginal dysbiosis. It's hard to tell, which comes first, because we've only know associative studies. So I've been working with Clive Gray and Tom Hope have a study in South Africa doing circumcision in men, and we're trying to add a component to that where we actually look at the women and BV and how that might alter the BV status. So that would be a good way to go about it, because we know that the microbiome is highly enhanced and you have less dysbiosis once you circumcise a man. And so would that be associated with decreased bacterial vaginosis? But it's a hard question to do. It's hard to get partners in a lot of these studies. But it is an important question, and I think needs further study, further research. Catherine Kripke, one of the side effects of the Depivorine vaginal ring is BV. Can you comment? I was not actually aware of that. I'm not surprised by it. I know the Nuva ring is associated with BV, and that is likely because so there's a couple things that have been hypothesized. One is you see biofilm formation. So when certain microbes, including Gardnerella vaginalis, which is the most highly associated with BV, can make these biofilms and basically trap the bugs in there. And then it's really hard to penetrate those with any good bugs. It's really hard to penetrate even with antibiotics. And I know biofilms can be formed on a vaginal ring, so that might be one mechanism. Also, if these dysbiotic bugs like to eat the Depivorine, and it's a food source for them, right? So this metabolism, they're doing it because they're using it as an energy source. And you see increased growth. So if they're eating the Depivorine, then these bad bugs might outgrow a good bug because they're using it as a food source. These are both conjecture. These have not been proven, but those are the two probably major mechanisms that I would think would underlie that increased BV effect. But again, those are just hypotheses that haven't been tested. Amanda says, then is it better to promote vaginal microbiome to enhance PrEP, not necessarily the gut microbiome? For women, and again, it's hard because we might be able to bypass this with either oral or the IM and injectables, but in general, not even so. The other thing that I didn't show, but so the vaginal microbiome very much influences inflammation. Well, McKinnon had a beautiful study, actually also using Capriso 04, showing that despite PrEP, inflammation actually overrode the effective PrEP. So if you have high inflammation, the PrEP was much less efficacious. So I do think that it's still really important, no matter what, to study that microbiome, because it's, even if it's not metabolizing the drug, even if it's bypassing it, using an injectable or doing oral, maybe it's not, we're not quite sure yet how much it's depleted by oral, but there's still this matter of dysbiosis in the vaginal microbiome causing inflammation and barrier damage that can cause increased HIV transmission. So no matter what, I think it's an extremely important thing to address. Judith Wolf, would you know how often OBGYNs monitor vaginal pH or Lactobacillus predominance in their patients on a routine basis, especially those at potential increased risk for HIV acquisition? I have no idea to the answer to that question, honestly. I would hope a lot, especially in the US, but I doubt it is. Even, at a root, unless you have symptoms of BV. Even, a routine OB/GYN exam in the US, you're not being tested for BV unless you have an active, I go in because you have symptoms. But I do think it's really important that we do better monitor, and I think it should be better monitored. And I think that it could really affect outcomes of overall STI and HIV, because it's not just HIV. I mean, it's all STIs have been associated in some way, shape, or form with BV. So again, anytime you have this increased inflammation, you have a higher risk of getting a pathogenic infection. Annie Elberry, do you know why IUD use is associated with increased BV? That's an interesting question. I do not know. I don't think that anyone knows the exact mechanism. I suspect it is a hormonal change, but I also think it's interesting because OB/GYNs love IUDs. Most girls I know are on IUDs, and it is constantly associated with BV. And so it's kind of mind-boggling to me how much they are recommended now. And I think it's very concerning. I do understand it's a really great birth control method that's long-term, but there are definite correlations between hormone levels, and especially local hormone levels, and different bad bugs. And estrogen tends to drive, like even in the gut, some negative dysbiosis. So my hypothesis, again, hasn't been proven, is that it would be a hormone-driven effect, but it's something that should be better studied for sure. So that looks like the end of the questions. If there's no furthers, then I will just say thank you all for your time. I love how many questions I got, and if anybody has anything following up, please feel free to reach out, and I hope everybody has a wonderful rest of your week. Thank you so much.
Thank you, Dr. Klatt. Evaluations and information on how to claim continuing education credits will be emailed by 5 p.m. Pacific time tomorrow, and this will enable us to review all of those that have attended today's live broadcast. The list of upcoming webinars available for sign up are on the IAS-USA website. Next week, we have a webinar with Dr. Marie Brown and Constance Benson, and that will be going over vaccine hesitancy and effective messaging for patients and healthcare teams. A new IAS-USA website feature, Practice Question of the Week, is now available to help our audience stay informed of the state-of-the-art practices for prevention, treatment, and care for people with HIV, COVID-19, or other viral infections. The practice question will appear as a pop-up when you visit the IAS-USA website, and you can also access the question through our resources tab. And save the date for the HIV/AIDS Ryan White clinical program. And for more information, please visit the website. Lastly, here's a list of recent on-demand dialogues. And again, we'd like to thank our presenter, Dr. Klatt, and for the audience for your participation. This concludes today's webinar.